Hello, folks. Welcome to section 6.6 in AP Biology, unit on gene expression and cell specialization. In this one, how do we specialize these different cells? It's pretty much just an extension of what we already learned about in gene expression, just specific to specialization. So a little bit of an overview here. So a program of differential gene expression leads to different cell types in multicellular organisms. Don't worry about this with single cell organisms, obviously. They don't have differentiated cells, they're all one cell. So, how does this happen? So, when you are developing the embryo, and the embryo comes shortly after fertilization, and it's when the cells start to differentiate. Before we get to the embryo stage, the cells really haven't differentiated yet. Okay, they're just reproducing. When they start to differentiate, that's when we say it is an embryo. Okay, so a fertilized egg gives rise to many different cell types. These cell types are organized successfully into different tissue types, then into different organs and organ systems in the whole organism. And gene expression orchestrates the developmental programs of these animals. So we turn on certain genes at certain times in certain cells, gives you, or a squirrel, or a worm, or a oak tree. All right. So the transformation from zygote to adult results from cell division. Obviously, you got to get more cells. You got to differentiate those cells into different types of tissues with different jobs. And then morphogenesis is making sure those cell types are in the right spot so you get the right structure, because morphology is the study of organismal structure. Well, I just said it here, but cell differentiation is the process by which cells become specialized in both structure and function, because those always go together. The physical processes that give rise to an organism's shape is what we call morphogenesis. Like I said, morphology is the study of that shape. Differential gene expression results from genes being regulated differently in each cell type, which we already touched on in the last section. And there are materials in the egg that set up gene regulation that is carried out as cells divide. So let's see how that happens. There are things called cytoplasmic determinants and inductive signals that we're going to look at. So in an egg cytoplasm, there are RNA, proteins, and other substances that are distributed unevenly in an unfertilized egg. So there's more of this RNA over here, more of that protein over here, more of this over there. It's not an even homogeneous mixture. It is uneven, for lack of a better term, but I can't think of anything better right now. Cytoplasmic determinants are maternal substances, maternal meaning mother, in the egg that influence that early development. So these substances that are unevenly spaced. As that zygote divides by mitosis, cells contain different cytoplasmic determinants, which lead to different cell expressions. Got a picture here. I'm just going to skip, I'll go back and forth. So here is that unfertilized egg, the nucleus, and we can see we got more of these round ones over here and more of these triangular green ones over here. So then we fertilize it. So then we got a fertilized egg, a zygote, and when it goes through the first mitotic division, if it divides right down here, you notice how this cell, this daughter cell, has a lot more of the round ones, and this one has a lot more of the green ones, and that starts to lead to some of that differentiation we were talking about, just because of the uneven distribution of these determinants in the egg. Okay. So that's what we're talking about here. Now, the next slide that I skipped before, the other important source of development information is the environment around the cell, especially signals from nearby embryonic cells. And we talked about this when we talked about cell-cell signaling earlier in the year, where cells can send out signals to other nearby cells to get them to do certain things. In the process called induction, signal molecules from embryonic cells cause transcriptional changes in nearby target cells, meaning we send a signal from this embryonic cell to its neighbor, and that causes this one to transcribe certain proteins and not others. Okay. Thus, the interactions between these cells induce differentiation of specialized cell types. So that's what's pictured over here. So here we got a very early embryo with 32 cells, and these cells are sending signals to these cells, their neighbors. So these cells are sending out these signals. We got a signal receptor, and then we got a transduction pathway that affects some change in gene expression in the nucleus. Here's a video that does a little bit of that to remind you of kind of what we talked about with cell-cell signaling earlier in the year. Catch you on this side.
Cell-to-cell signaling, with molecules carrying signals from sending cells to receiving or target cells, is a key mechanism in the coordination of cellular activities. In most cases, a signal molecule acts by binding to a receptor protein in the plasma membrane of the target cell and activating a series of changes, a signal transduction pathway, that triggers a specific response within the cell. A signal molecule binds to a specific receptor in the plasma membrane of the target cell. This activates a series of relay proteins, which in turn activates a transcription factor. The activated transcription factor attaches to the DNA and triggers transcription of a specific gene. The mRNA message is translated into a new protein, which changes the structure or function of the receiving cell.
And then, so there's how we kind of get cells, or partially how we get cells split up, but how do we get the different genes turned on at the right times? So what happens is determination irreversibly commits a cell to its final fate. It's like once you go through determination, you're going to be a liver, you're going to be a muscle, you're going to be a nerve. Okay. Determination precedes or comes before differentiation. So there's going to be some genetic determination, which then will lead to making certain proteins and RNAs, which lead to the cell becoming different. So genetic determination, differentiation happens once you got those proteins being produced, that specific tissue-specific protein. Just an example, I don't think you need to memorize this example or anything, but just an example. There are cells that are called myoblasts, which are cells that are destined to become muscle. Anytime you see myo, it has to do with muscle. MyoD is what we call a master regulatory gene, which has transcription factors that commit that cell to becoming skeletal muscle. So if you can turn MyoD on, then MyoD will turn on all the other genes that are going to turn it into a muscle cell. So here's just an image of that. So here we got an embryonic precursor cell. It's not a muscle. It's not a this or that yet. Here's the master regulatory gene. It is off, not being transcribed. The other muscle-specific genes also off. We have some sort of signal, whether it's induction or spatial, which causes us to turn on this gene. It starts to get transcribed and making mRNA, okay, and which causes it to make MyoD transcription factor, which then acts like a feedback loop to keep this gene on, the MyoD gene on, and turns on these other genes. Well, this gene, which turns on these genes, so we get a cascade here, right? This one turns on this one turns on both of these, and we wind up making the proteins that we need to make a skeletal muscle. Another thing that can be determination is pattern formation, is the development of spatial organization of tissues and organs. So when an embryo starts to form, we have this kind of like mass of cells, okay, and we establish major axes. Like this is where the head's going to be. This is where the feet's going to This is where the feet are going to be, and then things will develop off of that. So once you determine that initial axis, then you can decide what things are going to become what based on where they are in relation to this midline of the body. It's been studied a lot in fruit flies. Don't think you need to know that. Here's a video that kind of goes through it in fruit flies. I am not speaking well this morning. Fruit flies, and kind of talks about how this positional stuff works with a good visual. So I'll let you watch that. I think it does a better job explaining it than I just did.
Over the last few years, research has shown that mRNA molecules in the unfertilized egg of Drosophila determine the embryonic axes and future body plan of the fruit fly. The mRNA molecule that specifies the anterior-posterior head-tail axis of the embryo is transcribed from an egg polarity gene which codes for a protein called bicoid. This gene is active, mRNA is made, in the nurse cells that surround the developing Drosophila egg. The bicoid mRNA, once transcribed, is deposited into the Drosophila egg asymmetrically. More bicoid mRNA is deposited at one end of the egg than the other. Fertilization of the egg triggers the translation of the maternal bicoid mRNA into protein. The bicoid protein then diffuses through the egg, forming a gradient. The bicoid protein gradient establishes the polarity of the embryo along the anterior-posterior axis. The head and thorax will develop in the zones of high bicoid concentration. The abdomen will develop in the region of low bicoid concentration.
Last thing I want to mention is evolutionary development, or Evo-Devo. Down here we got this picture of a couple types of fruit flies, wild type and mutant. Okay, we got the eye, we got antenna growing out here. So normally these cells will get a signal to get the antenna genes turned on. But we've seen this where they actually grow legs out of where they're supposed to be growing antennae, which means that, because these cells, we know all the cells have the genes for making all the different parts. It's just a matter of whether you turn them on or not. So something in here, a mutation or something, caused the leg genes to get turned on here instead of the antenna genes. And so now this fly has legs growing out of its head. And that's a whole field of biology called Evo-Devo, or evolutionary development. And we think it's a big part of how we can gain parts or lose parts fairly quickly. We don't have to change all the little genes. We just have to change the master control genes for certain things, which is pretty cool. And that'll do it for this section, guys. Join me back for the next section on mutations. See you then.